This video is part 3 of Chapter 3, Sensation and Perception for PSY 110S, the Psychological Sciences. We're going to continue our exploration of the visual system by looking at the two types of visual receptor cells, the rods and the cones. Rods, which there are about 120 million of them in each eye, are principally responsible for our vision in dim light, and they are achromatic, meaning that they produce colorless vision. The rods are located mostly on the periphery of the retina. When you're looking at a scene, if you focus your eyes in one location, but kind of pay attention to the edges, you might be able to notice that the color fades. Now, not everybody can do this. It's particularly noticeable, the effectiveness of the rods, when it's darker, when it's dim. That's when color is not perceived particularly well. That's because the rods are the most likely to receive that information. So they're responsible for dim light vision, and what you see with the rods, what you perceive, is basically achromatic. This also means that your vision, as produced by the rods, is low acuity, means it's not highly focused. So if you're in dim light, you might recognize that objects appear fuzzy. That's because the rods have lower acuity than the cones. Cone cells are largely responsible for our color vision and for perception in bright light. There are about 6 million in each eye. They're located mainly in the fovea, that is the central portion of the retina. When we're in bright light, the cones are taking in the most important information, and they're responsible for our perception of color, or chromatic vision. They also have much higher visual acuity. So when you're looking straight at some object, like if you're looking at the letter C in the word cones, you'll notice that everything else that spreads out around that focal point is going to become more blurred. And the farther away, in speaking retinally, the farther away from that central point, the more blurry the vision gets. So we're kind of designed to perceive with the greatest acuity on that fovea where the cones are located. That gives us the most sharpness, the most nuance, the most color. Rods are really designed for helping us to navigate in dim light. So in normal bright light, the cones are mainly responsible for what we see. So in the daytime, when we're in bright light, our rods aren't doing a lot of the heavy lifting. That's really on the cones. So objects that we're attempting to look at are focused on the fovea, which is tightly packed with these cone cells. Cones provide a clear picture of objects because, especially the foveal cones, the cones that are right there on the fovea, tend to be more direct routes than direct routes to the visual centers than the way the rods and the bipolar and ganglion cells are located. Typically, one foveal cone is going to communicate with one bipolar cell, whereas for the rods, single rods can communicate, or rather, several rods can communicate with the same bipolar cell. Remember that there are a lot more rod cells than there are cone cells. Cone cells have direct lines to their own specific bipolar and ganglion cells, whereas for the rods, their connecting points are going to be shared instead. This means that the information that the rods send forward has basically been averaged, in a sense, across several positions, and that leads to a less clear picture, a more blurred picture going forward. In dim light, the focus is on the edges of the retina rather than on the fovea. The rods are more densely packed in those edges, pulling in some information, especially motion information, so that we can recognize things moving off in the periphery. But they're also a bit more attuned to picking up information that's interesting or necessary on the edges and in dim light. If you are wanting to be able to see, like if I get up in the middle of the night and I want to be able to see objects in my path, I have to remind myself to not try and focus directly on the objects that I'm looking for. Like, let's say there's a stray laundry basket left on my bedroom floor. If I'm thinking, oh, I really would rather not run into that, I'm going to want to look up, and then I'm more likely to catch the image of those objects in my path by the rods. If I try to look directly at the object I'm worried about, that's where the cones are tightly packed, so I'm highly unlikely to be able to see those objects. Dark adaptation is a process by which the rods and the cones, through chemical changes within the environment around them, become more sensitive to light when it's dim. You also have sort of the physical aspect. In dim light, in darkness, your eye, the pupil is going to dilate to allow in more light, but that's kind of a separate issue to the one being described here, where you have this internal chemical change, which is going to allow those cells to be more sensitive to limited amounts of light. The cones adapt very quickly to darkness. Within 5 to 10 minutes, you're going to get the best level of adaptation the cones are capable of giving you. The rods are going to adapt more slowly, and over that time, you will end up getting to your maximum ability to perceive. Color vision, there are several theories for helping to explain how we see color. Remember that you have the physical light that is entering the eye. Then there various levels of processing have to happen in order for you to recognize that what you're seeing is something of a particular color. Like right now, looking at my desk, there's this really awful looking lime green pen. So think about something colorful in your environment right now. How is it that you end up taking sort of that raw light that enters the eye, and then it turns it into things with color? So the trichromatic theory basically assumes that there are 3 types of cone cells that are designated to be the most responsive to different wavelengths of light, so short, medium, or long, that roughly correspond to different colors, blue, green, and red, respectively. The trichromatic theory explains the most common type of color blindness, in which red and green can't be separated. So, we'll come back to this here in a second as we continue to lay out this theory. The 3 types of cones have slightly different what's referred to as photopigment, which is, and these cells, based on their particular photopigment, they're going to be sensitive to blue, green, and red wavelengths. This assumes that many colors that we can perceive are mixtures of these various proportions. So, a light comes in, and the various the ways that they vary in terms of their wavelengths, they're all going to be a bit different. So let's say if it's a very, very bright red, those red cone cells are going to be highly active in sending their message. As that red becomes purple, you're going to have activation of different combinations of cells that are then going to produce a combined signal to represent those colors. If all three types of cones are equally active, what you're going to see is white. Proposed mixtures of primary colors are described as being additive mixtures. The different wavelengths of light are directly mixed together to produce the various variations in color that we see. This is different from how paint works. So if you're mixing paint, there are wavelengths that are absorbed or subtracted, and they don't get reflected from the mixture to the retina. So in the way we perceive light, it's an additive process. So you're combining wavelengths together directly to mix them. When we are looking at paints and mixing paints together, like if you take red paint and blue paint and mix those paints together, you end up with something purple. There's a subtraction process that's going on there. Light works in an additive way. Let's further explore this subtractive and additive mixture idea. In the first image, where you have a subtractive mixture, some wavelengths are absorbed or subtracted, and therefore they don't get reflected in the mixture. For example, mixing equal parts of red, green, and blue paint is a subtractive mixture. The intersection of all three colors appears to be black. In the second image, image B, mixing equal parts of red, green, and blue wavelengths of light is an additive mixture. So all of the wavelengths are directly mixed together, and that produces different results than mixing paints. So the intersection of all three colors of light is going to appear white as opposed to black. The binary additive mixtures, that can be, it seems very counterintuitive. So the additive mixture of equal portions of red and green wavelengths appear to be yellow, which, if you were playing with your paints, it ends up being this gross kind of brownish, orangish thing. Whereas in some cases, like the additive mixture of red and blue, you get some sort of magenta, whereas in the subtractive mixture, it appears to be more wholly purple. So sometimes these mixtures of colors appear to make sense. Other times they don't, because they seem counterintuitive, like red and green mixing to make yellow. A second theory about color vision is something called opponent process theory. Opponent process theory assumes that there are opponent process cell systems. So think about the words there, opponent process, meaning that they have opposite processes or processes that are opposed to one another in an interactive way. These help us see color in that they are located at the post-receptor cell. So after cone processing, so after you've gone through the cones, you have opponent process cells stimulated by, for instance, if it's a blue-yellow opponent process, you're going to have cells stimulated by blue light and inhibited by yellow light, and others that are stimulated by yellow light and inhibited by blue light in opposition to one another. So these systems are in pairs. There's red-green pairs and there are blue-yellow pairs, plus black and white pairs that are designed to help you see different levels of brightness. The colors inside each system oppose each other. If one is stimulated, the other is inhibited. Researchers have identified that there are ganglion cells as well as some cells in the thalamus that actually function in this way, following sort of the assumptions of opponent process theory. Opponent process theory can explain why we have certain kinds of after image effects. So if you stare at an image on a white background of the American flag, for example, and you stare at it for a good, 10-15 seconds, and then you stare at a blank white background, what you will see, instead of the red and white stripes, you'll see green stripes. Instead of the blue, you'll see the opposite in the opponent process. Now, there's also a composite theory, which combines the two theories we've already looked at, the trichromatic theory and the opponent process theory. This suggests that to fully understand color vision, you need both, because both correspond to different sort of physiological elements within the visual system. The trichromatic theory is most directly related to the cones themselves and their different sensitivities to different wavelengths of light. Then you have cells beyond that cone level that are aligned in opponent process forms. So in order to fully understand color vision, you need both. You need both theories because they're at different locations within the visual pathway. This helps explain why these theories are complementary. Color information is processed by the cones, according to trichromatic theory, but color information is further processed post-receptor by ganglion, bipolar, and thalamic, as well as cortical cells in an opponent process way. And that concludes part 3